I see that the recording's just started, so I will just reintroduce myself. Welcome everybody to the Safe Cassava Network. I'm Ros Gleadow from Melbourne, Australia, a plant scientist. With me Isabel Mendoza, who's the Chief Communications Officer of the Global Plant Council, and we're very thankful to the Global Plant Council for hosting this event. Also with us is Kira Ma, who is providing administrative support and will keep an eye on the chat during this session. And also thanks to the members of the Safe Cassava Working Group who have organised this symposium. A couple of quick housekeeping notices. First of all, feel free to use the chat, but you need to use the Q&A tool at the bottom of your screen to ask a question. So questions in the chat won't be answered by the speakers. To ask a question of the speaker, you need to use the Q&A tool. You can ask questions in the chat, but that will just be others answering that. There is an option to display closed captions in different translations with real-time translation or slightly delayed. To do this, you will see on the bottom of your screen there is a thing saying captions. I can't actually see it right now while I'm sharing. You go where it goes CC, there's an arrow there. You enable translation if you haven't already done this. You can select your language, and then there are options there on how you want to display this or whether you want to display it as a transcript, which will come up in the space normally where the chat comes up. Kira will post the instructions in the chat on how to do this. And with that, I will pass you straight on to the chair of the first session, Thorkild Tylleskar. Just a quick question before, something else before I say that is that if you're watching this on the recording on YouTube, there are other closed caption translation options there which you can use. Now, I will stop sharing my screen and pass over to our first chair, Thorkild.
Yeah, good morning, good afternoon, good evening, depending on where you are. It's a great pleasure for me to welcome you all to this seminar on Konzo, which is one of the diseases or illnesses that has been incriminated for due to the cassava toxicity. My name is Thorkild Tylleskar. I'm a pediatrician and professor at the University of Bergen in Norway, and I have worked closely with all these three colleagues that I am introducing here today. We'll start by Desiré Tshala-Katumbay, who is a neurologist or a neuropsychiatrist, actually, in the original form, with a PhD on this particular topic and a long-standing career at Oregon State University and working very closely with colleagues in DRC, the Democratic Republic of Congo, which is also his country of origin. Over to you, Desiré, please.
You are muted, Desiré.
Yeah, thank you very much, Tylleskar, and also thank you very much for the invitation to be a speaker on this particular symposium. Can you see my screen there?
Yes.
Oh, thank you. So, I've been invited to discuss this specific topic of cassava toxicity, and actually, which I will make sure that I make you understand that I feel more confident to talk about cassava neurotoxicity today. 30 years ago, working with Professor Tylleskar on the field in the Congo, I remember asking those specific questions. What exactly caused the disease? And I remember he told me it should be cassava. 50% maybe less confident that time. I feel now 90% more confident that cassava may be an issue here. So what is this specific condition that is called Konzo, especially for those who are not very familiar with? It's a condition that has a sudden onset of what is called a spastic paralysis, and you can see that illustration on the screen. In a formerly healthy person, within a week, a person gets paralyzed and can't walk. Few days later or few weeks later, the person can walk and display a spastic gait, or when the person can run, you can see that the person has some spastic features. And I can replay that again, because this is a very distinct and clear picture of that neurological disease. And I made it intentionally to play the same video that Tylleskar, who's chairing this session, has made 30 years ago. And I can confirm and say to you that 30 years later, it's the same conditions that have been seen around for the 30 years I've been involved with the specific conditions. So the disease in itself, it's for neurologists, it can be classified as a motor neuron disease, meaning that it's a disease that affects specifically the motor system. And on this specific particular slide, you have that illustrations, neurons that go from the motor cortex to the spinal cord and providing nerve conductions to nerve informations actually to arms or legs. Those are the specific neurons that are affected in these specific conditions. And clinically speaking, for neurologists, when you get that specific lesion within the motor system, you get the type of spastic gait that you just saw a few minutes ago. And basically for specific upper motor neuron disease, you have no sensory symptoms or no urinary symptoms. And this is in contrast with some other type of spastic paraparesis known in other parts of the world, including the Congo, such as HTLV1 associated myelopathy. So this is a very specific conditions. You have gait issues, spastic gait, no sensory symptoms, and no urinary symptoms. And you can also see on the left side, the posture. It's exactly one of the spastic stance that is known to be very present in spastic conditions. On top here, it's a condition that has been described, I will talk about this later on, more than 50 years ago. And down there, a kid with the specific conditions, and they can see the stance is about the same in the Congo, and down there, the same conditions in Mozambique. So apart from the motor symptoms, it's possible. Let me repeat that again. So the motor symptoms are the more distinct features that someone who has been familiar with Konzo would be, that would be easy to be identified. But on top of that, there may be some other type of symptoms, including some kind of sensation of electrical discharge in legs at the onset of the disease. It's possible that subjects present with vision issues, few subjects, less than 10%, eye movement disorders, but very commonly you can also see speech impairment of pseudo-Bilber type. I just want to say something here that on top of the spasticity that you can see in the legs, specifically in the legs, because the most common form of the disease, it's the paralysis of the legs, except in severe cases. On top of those specific motor symptoms, Most of the other symptoms are something that are also, or that can be described in motor neuron disease, but they are not present when it comes to the conditions lasting for few weeks or months. It's also possible, of course, in those same contexts, that you may have some other type of symptoms or some other type of issues. And my colleagues will discuss those. Espérance and Michael Boivin will talk about anxiety, depressive mood, and perhaps cognition deficit. The reason why I say perhaps cognition deficit is because over the course of these 30 years of research program, we have had some kind of suspicions when we did electrophysiological studies many years ago, showing EEG abnormalities, that it was possible that those kids may also have cognition issues. And of course, Michael Boivin will talk about that. But in contrast to the motor symptoms that are very clear, we still have a long time to go, as we still need to make sure that we characterize well those kind of motor cognitive deficits. And then on top of that, in contrast to the motor symptoms that are permanent and irreversible, Michael Boivin, I think, will discuss the issues of cognitive deficit that seem to be maybe not permanent or not progressive. And it's possible that some kids, through certain type of interventions, may get better. But I leave that to Michael Boivin, who's an expert in the field. But having said that, the most important feature that I want to emphasize here is the motor symptom features that you find in the disease. So that has been very clear. So what is the epidemiology and the risk factors? For centuries, you can see on top there, 1920s in the Belgium Congo, the condition has already been described. And of course, you got several studies that came along with the late Hans Rosling, Julie Cliff, Aulette, Tylleskar, Banea Mayambu, and then eventually 1996, 30 years ago, I got also involved with the research team. And at the time, I started actually thinking more about that and decided that I will go to spend my lifetime research career on the conditions. That is prevalent in sub-Saharan Africa, many different countries. The time I was working with Tylleskar, we used to talk about the DRC as being the most affected, which actually was the country that was the most affected even now. But you also have countries like Mozambique, Tanzania, Uganda, Central African Republic, Cameroon, and then lately, Zambia got also on the list. Mostly in poor remote area, among populations that rely on cassava as a staple. Children and women are more susceptible. The time we have been working on the condition 30 years ago, it was very seldom that we saw children under the age of 4 affected by the conditions. But in Kahemba, few years ago, 10 years ago, you can see on the right side, that small kid actually was 2 years of age. So it's possible that you get also kids that are less than 4 years of age that are affected. Sometimes you find familial clustering. And again, one of the features that is very important with the outbreak of Konzo, you get some kind of seasonal occurrence. It's not like over the year that you get outbreak anytime, but usually it's after the dry season, especially in the context of food and water insecurity. So we had another protocol to be able to quantify or to objectively assess the issue of food and water insecurity. And we showed, and those findings have been already published, at least for the water insecurity aspect of that. Food insecurity goes in hand with water insecurity in those areas. And in Kahemba, which is the most affected area of DRC, you have so many issues, not only related to issues related to access of water, but even when it comes to the quality of water, Kahemba has important issues. You will see the pH of drinking water around 4, so very acidic water. And that's the same water that people use when they start processing their cassava, including processing cassava in the river and so on. So acidic water has been an important issue. So something that I need to emphasize here, food and water insecurity, big risk factors. And we have been lately using machine learnings and using different algorithms, we have been able to come up with some predictors. The single and the most predictor, the top predictor of risk for disease outbreak comes out as the single questions in those specific area among the populations that rely on cassava as staple food. If you get a family that can say that on a scale of 1 to 10, what is the likelihood that in your own household, you may stay a day without food? In those specific conditions, that was the most important predictor of risk of getting Konzo. So if you get the family that say, last week, I spent a day or a night without having food, that family is at a very serious risk of the disease. So we are still working on the findings, and of course, publications and manuscript will be soon in the pipeline. So this was like the bulk of our research work for about 20 years now. But we didn't want to stop there because we understood epidemiology and the risk factor. We understood the issue of exposure. We know from previous studies and what we have been doing so far that cassava itself, harboring linamarin, once it is processed or digested, lead to exposure to hydrogen cyanide, which may be in terms be detoxified into thiocyanate, and in specific conditions, in other conditions, from thiocyanate, or from cyanide, I'm sorry, you may also detoxify thiocyanate into cyanate and other type of metabolite. So we had a research program with 3 distinct lines of research. We look at a biomarker of exposure. We look at biomarker of susceptibility, because you may be exposed to the same conditions. You have the issues of family clustering, but not all the kids develop the disease. But we also look at, of course, marker of damage. So it's 3 different levels of markers. And that brought us to a kind of diversification of our research portfolio. We have colleagues, and some of you may have met Matt Brambles, who have been working on the susceptibility, the exposure and susceptibility at the gut level, looking at the microbiome. And of course, it came out with some kind of scenarios that discuss issues of susceptibility in populations that has more beta-glycosylase bacteria in the gut flora. And then another scenario of protective mechanism in subject that who may have more rhodanase enrichment in the gut. So, but I'm not going to spend more time on that because it's going to discuss those maybe in future meetings. So from the gut flora, we moved into the bloodstream and look at in a more systemic level. So remember, for those who are familiar with isonacid toxicity, acetylation, with the acetylation issues, it's possible that the way people detoxify chemicals may also detect the type of susceptibility. And we look at for single polymorphism, genetic polymorphism in specific genes that code for the most important enzyme that have been involved in detoxification of cassava. Those are thiosulfate sulfate transferase, which is known as rhodanase, but also mercaptopyruvate sulfate transferase. We also look at SOD1, which is not an important component in terms of toxicity, but because there are also other kind of motor neuron disease, including amyotrophic lateral sclerosis, in which SOD1 mutation have been involved, and because we also found out a very high level of oxidative damage in the system, at least when it comes to the biology of Konzo, we're also interested in looking at SOD1 mutations. We found no conclusive findings. Of course, we did exome sequencing for all these genes, and there was no specific SNP that could be associated with increased risk for the disease. However, we found some other type of mutations, other type of mutations or SNPs in regions that are not coding regions, including regions that are involved in translational control, stability of protein, or subcellular localization of specific enzymes. But again, these were just indicative of what we found after carrying out the studies in a very small group of people. I would say that this does not indicate there is an issues of genetic susceptibility so far. Maybe there are some more studies that need to be done on a larger cohort of subjects. There are also other colleagues of us looking at the methylation pattern. We have issues of toxicity here that involve chemicals that can modify protein, meaning that can modify macromolecules. So in the search of biomarker of susceptibility and damage, it makes sense that we went around and look at also for epigenetics and find out that, of course, Konzo subjects have a different pattern of epigenetic profiling. So I talk about marker of exposure, well-known, susceptibility on top of nutrition or undernutrition. That is an important factor when it comes to the toxicity of cyanide. It's possible that the composition of the gut flora is an issue, and it's also possible that the metabolism, the metabolic rate, when it comes to the disease, to the susceptibility of disease may play a role. So we did look at the damage at the molecular level in those subjects. Few things that we have identified that make striking difference between Konzo and non-Konzo subject. You have a very high level of oxidative damage, as indicated by high level of isoprostane in plasma in Konzo subject, and the level of isoprostane seemed to correlate with the extent of deficit motor-wise or even cognitively speaking. And then in addition to the oxidative damage, you also have protein carbamylation that seems to be an important marker. Carbamylations actually arise from protein modifications by cyanate, which is also a metabolite of cyanide in those specific subjects. So as I said before, so we start with epidemiology 30 years ago, and then conducted additional studies on the epidemiology, including searching for risk factors, but also at the same time, we expand our research portfolio to assess other line of inquiries, including the marker of susceptibility and then marker of damage. So we conduct not only studies in human subjects, but we also conducted studies using experimental models, cell culture, rodents, and non-human primates. So we came up with a ton of informations that actually make us believe even more and be even more confident that the whole issues of Konzo is something that is related to the toxicity. We have shown a kind of strength or strong associations between the exposure itself, the metabolites that are the concentration in the metabolite that are produced when this subject detoxifies cyanide. And that correlation is actually with motor symptoms and cognitive deficit. Across different studies, experimental evidence came out that mostly thiocyanate and not cyanide, neither linamarin or cyanate, it mostly cyanate in experimental model that can induce a motor system disease. Cyanate, I'm talking about OCN, and the same cyanate also produce carbamylation in a human subject to the extent that level of protein, carbamylated protein, actually correlate positively with the extent of motor deficit. The more you are exposed, the more you seem to develop a more severe cases. Even experimentally speaking, of course, you get intoxicated with cyanide, you tend to develop more toxicity compared to those who are exposed to a lower dose. Coherence in findings is there from the epidemiology standpoint, the clinical standpoint, and experimental standpoint. You can see that we have a group of a ton of argument to say that it's possible that cyanide toxicity, especially neurotoxicity, is involved. Nutrition stay the same issues across the settings. It's plausible that the condition is induced by toxicity, and mostly, personally, my personal thinking, it's possible that cyanate, OCN, and not thiocyanate plays an important role, but because all these different chemicals, including cyanide, cyanate, can induce oxidative damage, nutritional deficiency can also induce oxidative damage, it's possible that there is not a single culprit, that most of the chemical may play a role, and this can be discussed when it comes to the susceptibility of the disease and why the subject would present mostly with the motor system. So this may be discussed later on or in a different symposium. And of course, a colleague of mine again, Matt Brembrew, is working on some kind on analysis that look at amino acid level, including cysteine, methionine, and then he also showed up in a paper that is published in Nature lately that glutathione peroxidase, GPX3, may be not only a marker of disease susceptibility, but also a marker of damage in Konzo. But I think he may discuss that maybe in a different symposium. What is the current theoretical framework for us? And why do we still have recurrence of Konzo in the Congo? Population continue to be exposed. And once you are exposed, especially at a very high level, we still have a marker of exposure in urines that we have been following up, extremely high, up to 1700 micromole per liter in urines. Stunting is still a big issue in those regions. And the same subject affected by the disease, when it comes to case control design, they have very high level of OCN, induced protein carbamylation, and that type of profile is mostly seen in subject with Konzo. So it's possible that once you develop the disease, and because you have issues with carbamylations related to OCN in your system, it's possible that you also have lower detoxification rate because most of the enzyme may also be carbamylated and lose functions. And that has been shown in experimental study in which treatment with OCN, with cyanate, carbamylate SOD1 and OCD1 loss of function was noted. And once you start having a very high level of oxidative damage and carbamylation, you detoxify low, and if you continue, if you stay exposed in the same regions, it's possible that there is a loop that get at risk people susceptible to the disease and even more being more affected, which means that some of the basis for the intervention that seem to be on the front line of what we've been doing Some of the bases actually are on this specific slide, and we have decided that we can try one specific aspect by reducing the exposure. It's an important issue. It's also possible that there may be strategy to enhance detoxifications through nutrition rehabilitation or some other things, but this will be in the future. We have shown that the odds of Konzo has been dictated by the cyanide detoxification rate. The more you detoxify, the more you detoxify, less you are susceptible. Malnutrition, as measured by stunting, is a risk factor, and of course, exposure to cyanide, high exposure, is also a risk factor. And we have started interventions with what we call enhanced weighting methods. The reason why I highlight enhanced weight method is because the weighting method that was put in place by late Howard Bradbury from the University of Australia, and we are celebrating actually all of this achievement today, I think, was used in some of the regions affected by Konzo in the Congo. And Pronanut has been able to show that after the weighting method, the risk for outbreak was substantially reduced in specific village. But we found out that the issues was sustainability. So weighting method by itself was sometimes not used anymore by those villagers, but instead, because of nutritional rehabilitation and advices provided to those specific village, so population start embracing different type of cassava processing, including diversifying their diet. So the intervention that we put 5 years ago in place in Kahemba to prevent Konzo use an enhanced weighting method, just meaning that the traditional method plus a package of nutrition rehabilitations. And we did that in a non-inferiority trial to show that using women themselves in the village, they can actually implement the interventions in a non-inferior manner compared to specialists. So study on Konzo, we have learned a lot on the specificity of the disease itself. There are few lines that can still be pursued there. Functional metagenomics, including microbiome studies that can open line on studies on pre and probiotics. Epigenetics, oxidative damage, lines on clinical trials. And of course, there are still things that are going on that we need to explore even more. Neurodevelopmental delay, neurocognitive deficit, and in adult populations in Kahemba, we have a very high rate, up to 30% of mild cognitive impairment. Likely, we had a rate of dementia that was comparable to other rural areas of Africa. But that was just based on the screening tools in which we show that the rate of dementia was about 13% in general populations. But a very high level of MCI can be an issue and need to be explored. It's possible that there may be some transgenerational effect that need to be explored. And of course, for those who are involved or we are fond of hot topics, climate variations and human health, cassava acts as a natural sensor when it comes to climate variations. Cassava are more toxic when it comes to drought. And of course, nowadays we use machine learning and artificial intelligence. So please, this is an opportunity to welcome you to the field of environmental neurology. We have been on the left side of the slide so far. Water stress, increased concentration of neurotoxicant, food and waterborne toxicant are there. This is an important paper that we published in Nature about 20 years ago. So, but we're still there. There are things that need to be explored. And of course, water and food security. It's an important issue. At the same time that we conduct research, we also build capacity, infrastructure-wise. We have samples, specimen, if you're interested, plasma, serum, DNA, urine stools, and we can ship around. We train, delegate power, empower students, and you can see on this slide, actually, a person there who is chairing this session has been with us for 30 years again. So thank you, Tylleskar, for your support and the support to the team. Several institutions have been participating, so we built global alliances. I can't just talk about institutions. I have colleagues who have been working with us, Espérance, Boivin, Michael Boivin for years now, Professor Mumba from INRB, Professor Okitundu from the Kinshasa School of Medicine, Department of Neurology, and so on, so many graduate students. And one of the big achievements, as I said before, we train, empower, and delegate from the field. Few years ago, many years ago, it was difficult, but we had the support of the Ministry of Health. I'm pleased to inform you that now Konzo is on the agenda, on the top subjects, 10 subjects on the agenda of the Ministry of Health, listed as Konzo, not as malnutrition, as Konzo. And then on the left side, one of the previous Minister of Higher Education was supported the team. And something that we may need to celebrate through this specific program. On the same slide, one of the graduate students at that moment, Therese, is now the Minister of Higher Education and Research Scientific in the Congo. And then on the left side, on the left side here, you have Okitundu, was also a graduate student on our program with the head of the Neurology Center in Kinshasa. And then we also have on the right side here, a special advisor to the Minister of Scientific Research in the Congo. So the team has worked tirelessly for the last 30 years. It's possible now that we'll get now Tylleskar grandchildren who need now to focus on the next step, and hopefully within the next few years, more light will be shed on not only the susceptibility issues, but also potential research venue for therapeutics or preventions. Thank you very much.
Thank you very much, Desiré, for this eloquent presentation of the current and past research that has been going on. And as you say, it has been going on for a long time also. There are a couple of questions in the Q&A. Desiré, you can have a look at them if you want to answer to them in writing or later. I'll give you a few minutes later to come back to the questions, so you have a chance to look at them. In the meantime, we will move on to Dr. Espérance Kashala, Abotnes.
So Thorkild, do you want to answer any of those specific questions for Desiré now, or do you want to just leave it all to the end?
I think you will leave it to the end.
Okay.
And then Desiré will also get the chance to read the questions.
Yeah, good, thanks. It was just such a great talk.
Yeah, so we move to Espérance Kashala Abotnes. She is also a neuropsychiatrist with special interest in children. And she has also several decades of research experience in this field. And she will talk about dietary cyanogen exposure and early child neurodevelopment, findings from the Democratic Republic of Congo. I should say also that Espérance is also originally from Congo, but currently at Children's Hospital of Eastern Ontario in Ottawa, Canada. Please, Espérance. You're on mute, Espérance.
Can you hear me now?
Yes.
Yeah. Okay. Thank you very much. Good afternoon, good morning, or good evening, depend on wherever you are. Thank you for joining me in this symposium. As mentioned, my name is Espérance Abotnes. I'm from the DRC. I have a position as an associate professor at the Department of Neurology in Kinshasa University, and also I work as a staff psychiatrist at the Children's Hospital of Eastern Ontario. For this presentation, we want to present on dietary cyanogen exposure from poorly processed cassava and its impact on early child neurodevelopment and also cognitive development. And this research mostly highlights the critical public health issue in cassava-dependent communities, and mostly Kayemba, where we have done the research. Let's dive in, and a quick overview of the findings and also the funding. So the comprehensive finding from the cross-sectional research which we conducted in Kayemba shows that we looked into how exposure to cyanogen, dietary cyanogen, can affect early neurodevelopment and cognition. This study was funded by the Research Council of Norway and NIH. Looking into the background, we can see that over 600 million people globally depend on bitter cassava as their main dietary staple. And in sub-Saharan Africa, it provides up to 37% of dietary energy, which average a consumption of 300 grams per person per day. This is a very critical public health challenge. And vulnerable population includes more than 200 million children below the age of 5 who fails to reach their developmental potential because of this dietary neurotoxicity. And geographically, hotspots like the Democratic Republic of Congo, where cassava is a staple food, has shown different in different studies how this exposure affects population. It has been done not only in the DRC, as Desiré has mentioned, but in other countries such as Mozambique, Tanzania, and even Nigeria. Our study was conducted in the Democratic Republic of Congo, in Kayemba, which you can see in red there. The region isolation, this part of the country is very isolated, and also it's a poor region. And that region relies mostly on cassava as a staple food. But unfortunately, the situation is very difficult because of the poorly processing. People process in a traditional way, making the population more at risk of being exposed to dietary cyanogen. On the top in this picture, you can see how local people are harvesting cassava tuber from the water, which is a very intensive and labor process. On the top right, you can see a very huge field of cassava, which emphasizes the role of cassava as a staple crop. And on the bottom left, you can see some of the uprooted tuber from cassava, which are rich in cyanogenic glucoside when they are not properly processed. And on the bottom right, you can see how the people in the community are preparing and washing the cassava to be eaten and transformed in different variety of food. Those pictures shows that the cassava can be transformed in a main staple food called the fufu, and they can even use it to make other pastries and different dishes. When it comes to the research method for our current study, what we did, the study was a cross-sectional study which involved 114 mother and children. We used a dyad. Those children were aged 12 to 48 months. And to assess them, we use essentially 3 assessment scales. The first one was the Mullen scale of early learning to assess cognition. We used the Gansini-Gavito scale, which assessed mostly the child early neurodevelopment as well as motor skills. And to assess the maternal well-being, we used the Hopkins symptom checklist to screen for maternal depression. When it comes to biomarker, we wanted to identify how is the exposure within those households. So we measure the cyanogen in the household cassava flour, as well as the thiocyanate contained in the urine of the mothers and the children. And then we conducted some statistical analysis. We employed multivariable generalized linear model with gamma function, adjusting mostly for socio-demographic and socioeconomic factors, as well as biological predictor. What did we find? The key findings from this multivariable analysis shows that there was a significant high level of dietary exposure in that population. And those were associated with almost all the outcome of the children, the cognition, the child development, and the fine motor. And we can see that there is a high clinical impact. The higher the exposure, the higher the clinical impact. And we also find that linear growth was a strong predictor. And as Desiré mentioned, those who had poor nutrition were more at risk of presenting deficit, cognitive deficit and impairment among those children. Some of the results we also found was that the level of maternal depression was quite high in the family with Konzo compared to those the control ones. We also noticed that the complementary feeding was introduced earlier in the Konzo household. As different factors associated with the child early neurodevelopment, we found the parental level of education and also the child anthropometric as mentioned. The linear growth was a strong predictor of child early cognition, motor functioning, and cognitive abilities. When looking into the exposure, as you can see here, the level was very high. When it comes to cassava flours, it was 5 times higher than what the WHO safety threshold. So 5 times. The W threshold is the 10 ppm, but in those families, the content in the cassava flour, which was used by the household, was 5 times higher. It was exactly the same when we looked into the urinary thiocyanate. It was also very high among children and among the mothers. You can see here for the mothers, they were around 800, while the children were around 600. And the WHO reference is around 300. So the mother had a level twice higher than the safe, acceptable or threshold from WHO, while the kids were also at twice higher. to summarize, we will see that those thresholds were extremely very high, and it shows that there is a need to do something within those communities. As you can see on the table, for the children, they were at 617, which is below the 350. Exactly the same for the mothers, and then for the flowers. In conclusion, based on our finding, what we can say is that the Konzo disease was associated with early child neurodevelopmental delays and cognitive impairment. There were some sociodemographic factors which were also associated, such as the parental level of education, the family wealth, the maternal depression, but mostly the child nutritional health, which was a major predictor of early child neurocognition and neurodevelopment in Konzo-affected areas. Those results show us that there is a need for further studies to see what can be the causality between those associations. Of course, we know that diet has a huge contribution. The exposure from the dietary cyanogen plays a big role, but we need to do further studies to find out what are the cause of those associations. Based on those findings, we thought that it is very important to have some intervention strategies for the population to improve their health. Some of the intervention which we found might be very helpful is, as Desiré mentioned, is the enhanced processing method, because it has been found that it will help to reduce the exposure from the staple food. Some agricultural intervention to promote biodiversity in the cassava can improve also the level of exposure from those populations. In addition to that, we also find that community education programs are very important. And as mentioned by Desiré, some of the mother were trained in addition to detoxifying the cassava, but using other methods such as nutritional, good nutrition to help those families to reduce their exposure to those dietary cyanogen. Neurorehabilitation services are also very important, and there are different interventions that can be done with those children to improve their cognition and their early development. And I think Professor Boivin will speak about some of the intervention which we have put in place to promote children cognition and early neurodevelopment. What are the challenges? There are a lot of challenges in Kahemba. First of all, the access to the place is quite challenging, but we have also the environment. Okay, there is poverty, there is the soil, which is very acidic. So there are a lot of situations within Kahemba which are quite challenging. But we know that the wet processing methods are low cost, and those interventions can be scalable and suitable for resource-limited setting. So early intervention programs such as the MISC for those affected children will help to improve their cognition, their motor development, and mitigate different associated risk factors. Thank you very much for your attention, and please, if you have questions, I will be very pleased to answer them.
Thank you very much, Espérance. Please post the questions in the Q&A, and then we will have some time at the end for remaining questions.
Yeah.
So it is now a great pleasure for me to introduce Michael Boivin, who is a University Distinguished Professor and Research Director of Psychiatry and Professor of Neurology and Ophthalmology at the Michigan State University in the US. And he has spent many decades, I would say, almost 50 years in Africa, in different settings, and studied development of children with different conditions, HIV, severe cerebral malaria, and Konzo. I think his first visit to Congo was in the 80s, if I'm not mistaken. You are going to talk to us about the mother and child's health in response to early child development interventions. Please, Michael Boivin.
Hey, thank you. I'm gonna try to. I've got a lot of slides to cover. I'll only be able to get through a portion of those, but I will make all the slides available as a handout to Isabel and the other meeting organizers following this meeting, and they can be shared upon request. I'm going to focus on our most recent work. We're just finishing the second year of a 5-year study sponsored by the National Institute of Mental Health, where we're going back and looking now at the cognitive mental health and neurodevelopmental outcomes for children and their mothers who receive the earlier behavioral MISC, mediational intervention for sensitizing caregivers, training when these were very young children, just beginning to be weaned from breast milk to cassava porridge, training the mothers in ways in which we hoped would help foster greater bonding, better neurodevelopmental outcomes for these children, as well as a way to sustain the labor-intensive types of wetting method training that they were getting, the enhanced wetting method training. Again, a technique developed by Bradbury, validated in the Congo by Bania and Bradbury in collaboration with many other colleagues with support from PRONANU, the Ministry of Health. So we received a grant to now go back and look at mental health and behavioral benefits of this early caregiver training in the hopes of better understanding what factors help mothers bond to their children in early childhood and help sustain the labor-intensive needs of safer and better nutritional provision for their children and the entire family as these children get older and reach the middle childhood and school age. These are our basic study aims for the present 5-year research program sponsored by the National Institute of Mental Health. Again, we're looking at what factors related to this behavioral intervention of early caregiver training. What we've used are the MISC intervention, which I'll explain more shortly, and ways in which those behavioral factors, particularly the emotional well-being, psychosocial factors for mother and child, strengthened early on in caregiver training, might help the mothers prepare for safer nutrition and well as prevent Konzo over the long term. As Desiré and as Espérance have shared, our study site is Kihemba, which has one of the highest incidents of Konzo disease in the world, largely because of the movement of the population back and forth across the Angolan frontier in response to civil war, economic hardships, nutritional and water insecurity, and has really led to the population in Kihemba being dependent on bitter cassava but at the same time with few resources for other types of nutritional diversification. Now the MISC, the Mediational Intervention for Sensitizing Caregivers. We first used this with moms living with HIV in Uganda. It was an intervention strategy devised by Professor Nina Klein, who's now passed away, but who won the Israel Prize, their counterpart to the Nobel Prize, for her lifetime contributions in child development interventions, and really has used this particular strategy, which is really very similar to what UNICEF, WHO, their nurturing child strategies, which combine early caregiver training to enrich the home environment, along with nutritional enhancement and diversification. This is a bit of a busy model of MISC intervention, but really at the core of it, within the red square here in the middle, are elements of emotional enhancement, positive emotional experience, which enable the mother through weekly or biweekly training to use everyday interactions with the child within the household at a very early age to strengthen the neurocognitive skills and development, and over the long term, the emotional well-being of the child. And we have found with our work with moms living with HIV and children exposed and infected to HIV in their very early years within Uganda, that the children's neurodevelopmental outcomes and psychosocial adjustment is improved as well. So the mothers in their daily interactions, whether it's joint shared work or even play, washing, bathing, feeding the child, the mother learns through videotape systematic training by a peer-type trainer, somebody who can work with the mother in her home environment on a weekly or biweekly basis, how to utilize those interactions for ways to enhance the neural development of the child. And we have found that in the process of doing so, both mortality and morbidity were improved significantly compared to treatment as usual or non-MISC intervention. And at the same time that the children neurodevelopment and emotional well-being over the long term was significantly enhanced for mothers who received this kind of training over the course of a year in the case of our Ugandan interventions, as well as our preliminary studies in Kehamba as well, conducted by two different studies, one led by Espérance, funded by the Saving Brains and Foundation in cooperation with the Canadian Ministry of Health, as well as an NIH-sponsored R21, or a preliminary study by the National Institute of Child Health and Development of the NIH. And that laid really the foundation for the present 5-year research program, which involves following up with our children now that they're reaching school age. So we published a number of early findings, established the importance of this kind of early caregiver training. And the thought was, well, maybe if we do this kind of caregiver training with the moms in Kehamba, particularly at risk for Konzo disease in their households, then maybe this would help fortify their willingness and ability to sustain the wetting method techniques that they were learning, through Pronanu, to make the cassava safer within their households. As Desiré mentioned in his presentation in his non-inferiority trial sponsored by the NIH, that we can train the moms successfully in Kehamba to use the wetting method to detoxify the cassava flour up to 98% through wetting and evaporation and detoxification. But it's a very labor-intense method on top of already very difficult and daily demands on their day-to-day activities of daily care and living, gathering wood, processing cassava, providing, everything is labor intensive within these kinds of environments. And so adding the wetting method for all of the food that they need to prepare from the stored cassava flour can be a non-sustainable demand. And we were hoping to see if it can improve mortality and morbidity among HIV moms in Uganda by training Konzo at-risk mothers within the Kahemba households, would they be better able to sustain the demands, at least enhance or improve the development of their young children as they're weaned and on through early childhood into their school age years. And so in Kikwit, we trained a number of community caregivers who were leaders, women who were leaders within their communities back in Kahemba. We did the training in Kikwit initially using two of our very best MIS trainers from Uganda who were willing to come and help basically transplant south to south our MISC intervention. Fiona and Miriam, who were our two very best trainers from our Ugandan project with moms living with HIV, and in cooperation with Espérance, her support and translation ability, we were able to train our first graduate class of mother and community leaders in the use of the wetting method who could go back to their communities in Kahemba and then work with mothers in small group or individual settings on a weekly or bi-weekly basis, showing them and using videotape recordings on tablets, the techniques involved in ways in which they could enhance the development of their child in home-to-home environments of daily living. Half of our mothers within this clinical trial received MISC intervention in addition to the wetting method that was part of Professor Desiré's R01 program. And then half of them received only the wetting method so that we could compare the long-term neurodevelopmental as well as psychosocial, mental health, and Konzo prevention benefits of each, at the same time measuring toxicity within the cassava flour as well as urinary thiocyanate and other biomarkers of Konzo risk that already had been well developed and documented by within Professor Desiré's research program. So here we see one of our MIS trainers working with the household in terms of explaining the basic principles of MISC and ways in which they could enhance the development of their children day to day within their interactions with their children in the home environment. And then using recorded interactions of mothers bathing their children, feeding their children, working with their children, playing with their children, using those videotaped interactions that actually recorded with the mother and child to then later on show them opportunities for ways in which they could use MISC principles to enhance the caregiving environment for that child. And then again, the task was to see whether adding MISC could improve adherence to the wetting method as measured by cassava flour toxicity as well as glucogenic cyanide. We did measure a whole lot of other factors within our preliminary studies, both Espérance's Saving Brain study as well as my R21 study. We did see significant improvements, but especially in the area of mental health and psychosocial adjustment on the part of the child, as evidenced by the mother's responses to the Strengths and Difficulties questionnaire, the Jorgensen and other kinds of neurodevelopmental indicators. So we went back, selected as many of those moms and children that we could find, and now came back 3, 4, even 5 years later over the last 2 years and are looking at those mental health outcomes of children, both those that received MISC and those that did not receive MISC, but only the wetting method. All the children and moms received the wetting method, but again, through these randomized controlled trial preliminary studies, half did receive MISC. In addition to that, half did not. I'm going to move quickly. We've looked at some of the questionnaires. that the National Institute of Mental Health requires as part of any of their studies. We're now have 2 rounds of assessments on questionnaires, and we've seen preliminary evidence in support of the improvements in psychosocial adjustment from the early MISC intervention. Those findings, though, need to be confirmed and then will be presented in subsequent opportunities. But the other portion of the study was eye tracking. We were able to pioneer the use of eye tracking with Tobii eye tracking technology to look at responses of the children to various types of images on the screen, both those measuring attention to animated cartoons, like our early childhood vigilance test, which we found to be a very sensitive measure of attention and cognitive attentiveness that predicted then their cognitive abilities once they reach school age. But we also developed pictures that we could present to the child, which were very emotional, either frightening or else happy or even spiritual, as well as neutral pictures. And the eye tracking technology could give us a window into the brain by pupillary dilation in response to the parasympathetic nervous system emotional activation. We could perhaps gain a window into the brain, typically only available through imaging sources. And our preliminary studies over the last year in Uganda with adolescents living with HIV found that their eye tracking responses to these types of different spiritual versus happy versus sad versus neutral pictures result in very different types of eye tracking and emotional responses as gauged by their pupillary diameter and changes. We're also using a 6-minute video of mother-child interactions, a standardized video in which depicts the kind of MISC interactions in the village household setting that we had used in Uganda and then more recently in the Congo, and looking at what are the eye tracking responses to these video interactions. Does the child look at the mother, look at the child? Does the child especially drawn to interactions during times of the child's distress, during times of positive encouragement? And this is, we've seen very strong effects in our preliminary studies with both emotional images as well as videos. And so now we came back in August finally when we were allowed to because of the Security 4 level restrictions that my university and the State Department had in place for the Congo. We were finally able to come in, train our study team at INAB. These are psychiatry faculty from CNPP, University of Kinshasa Medical School. They then are graduates of that eye tracking technology training program, then left for Kahamba and returned just a few weeks ago. We were able to then, with the help of Professor Tshala, get the video files of the eye tracking assessments, which are very large and data intensive, to our neurocognitive laboratory colleague in the College of Human Medicine at Michigan State, who developed these tests and the Tobii eye tracking software to measure these. And then she's now, just as of last week, is looking at the quality of those videos and beginning preliminary analyses of what we think to be the very first eye tracking assessments ever to come out of Central or Western Africa. And the resolution of these are such that we can even pick up early neurological signs of Konzo disease risk in preclinical children in the form of nystagmus and voluntary tremors and so on. So it's a very, very sensitive technology, which we hope will provide an additional window into the mental health and neurodevelopmental outcomes of both early toxic cassava exposure as well as an early caregiver training intervention to prevent such exposures, such as MISC. And with that,
Bye-bye.
That's our team leaving Inari for Kehemba in anticipation of 2 very, very difficult and long days of travel, especially day 2 from Kikwit to the very Angolan frontier of Kehemba. And with that, I want to thank you.
Thank you very much, Michael, for this very interesting presentation. Thank you so much. We now have a couple of minutes for questions. I can see there is one question in the Q&A. I think perhaps Desiré, you could have a go on this. Could cyanide levels in pregnant women be transferred to the embryo or to the fetus? Desiré.
Yes, so okay, so of course we do not have evidence of that, but it's possible. And I'll talk more about metabolite, including thiocyanate and cyanate, which can readily cross tissues. So it's possible.
Ros, you raised your hand. Do you have a question?
Yes, I raised my hand because as panelists, we don't get access to the Q&A function, and I just really enjoyed these talks so much. So I do have several questions. So Desiré, you talked about how previous exposure could make you more susceptible to later Konzo. I was wondering, what do you mean, over a short period of time or over your lifetime? So if you have a lot of cyanates in your diet, can you expand on that, please? What's the effect of long-term or previous exposure? Are there cumulative effects?
Yes, so that's a good question, because that's exactly what has been consistent across Konzo areas. So outbreak occur when there is a peak in exposure, but actually on a bed of chronic and low-level exposure. So there are still things that we need to understand. Why do you have the outbreak and the sudden onset of paralysis when you have a peak? That is one question to be answered. It's possible that when you have a peak, then you trigger apoptotic mechanism that makes the disease, not the motor neurons to die and make the disease permanent and irreversible. But it's also possible, based at least on experimental insight, that long-term exposure, and depending on the type of adaptive mechanism that one may have, you may see people with different metabolism pattern, and including those who can maybe produce more of the thiocyanate metabolite compared to those who can produce more of the cyanate metabolite, which makes things, toxicologically speaking, that the phenotype of the disease may also depend on the type and the extent of exposure. You get very high exposure in cyanide, you die. You get low level of exposure of cyanide, you adapt and start developing metabolic issues. You get a peak on top of a low level and chronic exposure, you trigger neuronal death and you develop a disease. You have a prominent metabolic pattern based on cyanide. You develop a Parkinson's disease. Based on thiocyanate, you develop thyroid disease, and based on cyanate, maybe you develop a motor system disease. So there are still things that need to be learned of, toxicologically speaking.
Ros, you had several questions.
Oh, yes, I have several questions. So I have a question for Espérance, and I think Michael's talk sort of answered in some ways. Do you think, when you did that work on depression, and mental health in mothers. Do you think that depressed mothers are going to find processing more difficult and so higher exposure to cyanate, or do you think the exposure to cyanate may be linked somehow to the depression?
Yeah, that's a very good question because I mean we know depression can be multifactorial, but what we have found in this community is that the mothers in family affected by Konzo reported more symptoms of depression than the mothers still in the community but not affected by Konzo. So it shows us that mothers in Konzo affected families are more vulnerable to depression because it's a disease which affects not only, as we could see on the video, the physical health, but also the mental health. It has a huge burden on the family. So that's yeah, but it will be interesting to continue doing more research and see if the exposure are the one causing depression. But I'm sure it is multifactorial.
And I just wanted to add to that. In our work with moms living with HIV and MISC intervention in Uganda, we saw that depression was really an important modifier as to how effective the MISC training was in the ability of the moms to then adapt and utilize MISC. And our initial look at those data, at least from our R21 study, with MISC intervention in really early childhood in Kahamba, shows some of the same important modifying effects. So what we have proposed, not yet in the Congo, but what would be a really nice follow-up would be to combine MISC with a therapeutic intervention for the moms to address the greater likelihood of depression, and then see if that can affect or improve the uptake of both wetting method as well as the strategies of enhancing the neural development of the children in their early caregiving environment.
And if I need to add something, something that is clearly so far seen over the last 30 years and based on the longitudinal findings, what we have been able to show so far is that the motor symptoms are the key feature. And those findings are reproducible. The neurons die and you get the paralysis. And then on top of that, you have associated symptoms that you get in those areas that will need to be worked out again, and longitudinally speaking, understand the dynamic of those changes, if they may also related to the exposure, malnutrition, the disease condition, or anything else. So we have a clear picture of the disease, and we have additional issues in the area, and those one will take time to be worked out.
Thorkild, you're muted.
Thorkild, you're muted.
I'm sorry. Thank you, all three speakers. We have come to the end of this session. So thank you for your presentations, and I'll hand over to Ros for a final word. You are also mute.
Thank you, everybody, for attending this session. What I wanted to do, sorry, I've just got a problem here. Just give me a second. I've just, for some reason, something's happened. Okay, righto. So I think the first thing, thank you, Thorkild, for organising it. Thank you for those speakers. I think we need to also have a very big thank you to, before I come to this slide, a very quick big thank you to those who have gone before us and have mentored us. Howard Bradbury with the wetting method, Hans Rosling, Julie Cliff for their pioneering work in Mozambique, Jean Bernier for his work in DRC. Of those great giants, you know, Howard, Hans, and Jean have all, and Bernier have all passed away. So I think we have a lot to be thankful for these people, and I'm really appreciative of people giving their time to speak today. When Howard Bradbury died, he set up a Konzo prevention fund at the Australian National University. The symposium is free, but if you would like to give a donation, you can donate to that fund. And in fact, we're using some of the funds from that, a small portion of that, to just facilitate the funding to get this, the small amount needed to facilitate this session. So there's a QR code if you want to do that. No obligation at all to pay anything. So also, I just want to, now my screen has disappeared. Has anybody else got these slides? Okay, I just wanted just one more thing before I go on, before I let you all go and have a break, is that we also have next coming up, we have the cassava safety in the session with about cassava the plant, chaired by Carlos Iglesias, and that will be in 30 minutes time. So you've got time now to go and get the appropriate beverage or food for this time of day in whatever time zone you're in. So I'll see you soon. International Safe Cassava Network Symposium on Safe Cassava, which is hosted by the Global Plant Council. I am Ros Gleadow, and I'm a plant biologist from Monash University in Melbourne in Australia. And I study cyanide in plants, and that's my connection. And I chair the network and also put together the first steps of the International Safe Cassava Working Group. I want to acknowledge the members of that group that have put this symposium together. So as I said, with me, we have Isabel Mendoza, who is the Chief Communications Officer for the Global Plant Council. And we're really appreciative of the Global Plant Council hosting this session for us. It's a great thing, and the session will be recorded and posted on their YouTube channel afterwards. Also with us is Kira Ma. Kira is providing administrative support to the Safe Cassava Working Group and Network as part of the Konzo Prevention Fund. Some quick housekeeping notices before we start the session. Please feel free to use the chat. You can introduce yourself and talk, ask questions of each other, but to ask questions of the speakers, we ask that you use the Q&A tool. So if you see the toolbar at the bottom, it says Q&A. You just click on that and you type in your question and the speakers can answer it. I also want to alert you to the function that we've added where you can display closed captions or subtitles in the language of your choice. We recognise that cassava is grown throughout the world, and not all of the experts or all of the people interested are going to speak English, although most of our speakers, but not all, over the 2 days will be presenting in English. To activate that, you click on the up arrow next to the show captions at the bottom of your screen if you're on Zoom, enable the translation, and then select the language of your choice. There are options on how you want to display that, and also there's a possibility of it coming as a transcript where the chat would normally show on your screen. There is a slight delay in the transcript coming up, but if you prefer, you can have those coming, closed captions. If you are watching this on a recording on YouTube, there are other options there for real-time translation. So at this time, I want to pass over to the chair of the next session. I'm just going to introduce him. Is Carlos Iglesias. This session is of particular interest to me because I'm a plant biologist, and this is the focus of this session is on plant biology. So I pass over now to Carlos, who will introduce the speakers.
Thank you so much, Ros. I'm Carlos Iglesias from the campus of North Carolina State University here in Raleigh. And the session is about cassava safety, as Ros was saying. Then cassava has been a highly productive and resilient crop in the tropics. It can be toxic under certain circumstances, particularly processing. It contains cyanogenic glucosides, which can release toxic cyanide, leading to some health concerns. So this session deals with approaches to make cassava safer from the breeding perspective and from the biotechnology and gene editing perspective. Our first speaker is Dr. Nicolas Karavolias. He currently serves as a life science research fellow at the University of California in Berkeley, and he's an instructor with the African Plant Breeding Academy's CRISPR courses. He specializes in CRISPR-based editing in crops for climate change adaptation with experience working with rice, tomato, and cassava. Nicolas, the floor is yours. You have 20 minutes or less to deliver your presentation, and thank you so much.
You need to unmute,
There it
Nicholas.
goes. Hi there, everyone.
That's
Hear me
great.
all right? Awesome. Well, I want to say firstly, thank you to the organizers of this really important meeting. It's fantastic to be here, and I'm really grateful to be a part of this community that's working to address an important issue. We at the Innovative Genomics Institute at UC Berkeley are curious about the ways that we can use genome editing to be able to impart meaningful traits or characteristics in plant phenotypes, as well as in human health and many other aspects of biology. And so I'm here today serving as an ambassador for the work in cassava that we've done at the Innovative Genomics Institute. And I'll take you through a couple stories regarding how we've employed genome editing in the cassava crop to impart some meaningful phenotypes. But before I hop into the cassava work, I want to give a brief background on CRISPR. So CRISPR stands for clustered, regularly interspaced, short palindromic repeats. And it's not important that you understand what that acronym means so much as it's important to understand that this is a tool for precise genome editing. So what you see here is a cartoon of a CRISPR, a Cas9 module editing DNA. So what you see depicted in the green is a Cas9 enzyme. And this is a nuclease which has the capacity to make double-stranded breaks in the genome. And we, the researchers, are able to program where those breaks occur through the use of a guide RNA, as depicted in yellow. So this is a small piece of 20 nucleotide guide RNA that allows us to make directed edits in the genome of organisms that we're interested in working on, in this case, cassava. So what a researcher like myself can do is engineer these short strands of DNA to go into the genomes and create double-stranded breaks in the organisms that we're working with. And as a result of cleaving the DNA at a guide designated position, we can introduce mutations to the genome at that position. And so this has been a revolutionary tool for many domains of biology, from plant genome editing all the way through human biology. And we were really excited to be able to start working with this tool in the context of many crops and traits. And so just to accentuate the point a bit further about how CRISPR works, we introduce a guide sequence. The Cas9 molecule will introduce double-stranded breaks, and then a cell can repair those breaks. Typically, we're looking at the pathway on the left here, which is non-homologous end joining. And so you'll see that through the double-stranded break repair, we can get small insertions or deletions into the genes that we're interested in working with. And as a result, interrupt those gene functions, in our case, to produce knockouts. And these knockouts may be useful. So removing the function of specific genes, in this case, cassava, in order to generate some type of useful phenotype. And so that's exactly what we did. And I'm going to tell you a couple stories about how we've employed genome editing in the cassava crop with specific interest in influencing cyanogenic glucosides. So when we have these CRISPR constructs, we develop the guides, we develop the proteins that we want to work with. We actually have to deliver all this machinery into the cassava plant. And so you can see here a depiction of the steps that it takes to be able to go from a CRISPR concept, the desire to edit a specific gene in the genome of cassava, through actually regenerating an edited plant that can be used for characterizing and hopefully eventually being deployed as a improved variety. So we start with callus tissue, and that's what you see in A. These are clusters of cells that have the ability to be regenerated into whole plantlets. And we move it through this process of tissue culture, wherein we introduce this bacterium called agrobacterium that can deliver the editing reagents into the cells of our cassava plant. And then we select for the cells that have been transformed with that DNA that will allow for the editing and regenerate whole plantlets from those edited cells. So I wanted to give a bit of background about how the CRISPR technology works and how we might actually be able to generate whole edited cassava plantlets. And so we're very grateful to Taylor et al., the Danforth Center, for developing this method. And this has been really instrumental for our ability to do CRISPR genome editing. And with that, I'll transition into what we've actually done. So employing this type of protocol, we first embarked on a journey to use genome editing for cassava trait of viral resistance. So the first story that we told at the Innovative Genomics Institute was about using CRISPR to edit and improve the resistance of cassava to cassava brown streak virus. And this was ultimately quite successful. You can see some of the data as a result of this project. You can see that there are lower titer volumes of the viral load overall, and that these cassava plants were doing much better. And so this was our first proof of concept that we could actually employ the genome editing technique in cassava in our own hands and impart really meaningful trait phenotypes in the cassava crop. And so naturally, we became curious about cyanogenic glycosides. We saw that we could impart a meaningful phenotype for brown streak virus, but we wanted to also, having been inspired by the work of the field in characterizing the incidence of Konzo and the really deleterious impacts of cyanide in cassava, we sought to understand if genome editing could be a useful approach for addressing this issue. And so we were also very grateful to have the preliminary research or the prior research that guided us towards potential good targets for genome editing. So we were benefited from the knowledge of the biosynthetic pathway that converts valine and isoleucine to primary metabolites, central metabolites, to linamarin and litostrolin, which are the primary sources of cyanide. About 90% of all cassava cyanide is in the form of linamarin. And so it was great that we had the knowledge as to how these compounds or metabolites were being synthesized. And we thought, looking at the first committed step of cyanogenic glycoside biosynthesis would be a great place to begin to target in a genome editing strategy. And so we looked at CYP79D1 and D2 and thought about introducing CRISPR-Cas9 edits to these genes in order to potentially remove the function of these biosynthesis pathway enzymes. And so that's exactly what we did. We introduced guides or designed guides for both of these genes, both in tandem and separately. So we were able to make edits to each of these genes individually and also target them simultaneously. And you can see here, we had a total of 4 guides. These guides were designated in the first exon of both of these genes in the hopes that we could affect some type of small insertions or deletions through genome editing that would remove the function of these genes and as a result have some type of impact on the accumulation of cyanide in cassava. And sure enough, you can see our genotyping was a success and that we were able to validate that we introduced mutations into the cassava plants through CRISPR editing. And so you'll see here some of the edits that are designated in red, and you can see that some of them resulted in frame shift mutations, indicating that the open reading frame of the gene was shifted and everything downstream from that edit would, as a result, have a different amino acid sequence and also resulted, I can tell you, in every single case of a frame shift, introduced early stop codon. So we're really truncating these proteins and pretty confident that we had removed the function of these proteins as a result of our editing approaches. And what was really exciting is that we were able to generate edits in each of the genes individually that removed their function and simultaneously. And so naturally, we wanted to ask the question as to whether these genotypes had any corresponding impact on the actual phenotype of the plant, which in this case would be the accumulation of cyanogenic glycosides or the precursors of cyanide. And so we started in one cultivated variety, 60444, and we measured the volume of hydrogen cyanide per kilogram of tissue. And what we found is that in our double knockouts, meaning the edited events that had eradicated the function of both CYP79D1 and D2, we virtually had no cyanide present. So we weren't able to detect any cyanide in those plants. And what was really also quite exciting is that we could learn a bit about the relative function of CYP79D1 and D2. So what we found was that in the knockout background of CYP79D2, there was very low levels of cyanide relative to the wild type, whereas when we had the functional copy of CYP79D2 and no functional CYP79D1, there were higher relative levels. So this indicated to us that there was kind of unequal participation of CYP79D1 and D2 in the production of these cyanogenic glycosides. That CYP79D2 was actually perhaps a more significant enzymatic character in the story or the biosynthesis of cyanogenic glycosides. And so not only could the double knockout virtually eradicate cyanogenic glycosides, but we also had these kind of intermediate or tunable characteristics of the single gene knockouts from each the CYP79D1 and D2 knockout. So this was in 60444, but we repeated this work in 2 other cultivars, TME419 and TMS910234. And we found something similar across 3 cultivars. The double knockout in every single case resulted in virtually no detectable hydrogen cyanide. And in every single cultivar, we also see this outsized role of CYP79D2 plays in conferring more of that biosynthesis production, thereby in knockouts of CYP79D2, you see even lower levels of cyanide relative to just the single CYP79D1 knockout. And I think this is really notable because you can see that now we have a couple genome editing strategies in producing kind of a phenotypic array of cyanogenic glycoside content. If you seek to eradicate CGs, you can simply produce a double knockout. If you want very low levels, but not a complete eradication, you might pursue a single gene knockout of CYP79D2. And CYP79D1 as a knockout kind of was inconsistent across the cultivars of how that response would occur. So sometimes that CYP79D1 had near wild type levels, and in other cases had more of an intermediate phenotype. So it was great that we were able to explore these characteristics of genome edited cassava plants across many cultivated varieties and show that through the production of a double knockout, we can really more or less eradicate the accumulation of cyanogenic glycosides. And I just want to give a nod to some work that was happening simultaneous with our own by the folks at Puana University. Juma et al. Reported something similar in a CYP79D1 knockout. And so I just want to acknowledge that they found something quite similar. They report here that this edited event is CYP79D1 knockout, and they provide the guide sequence in purple here. I just want to note that this guide sequence is also present in CYP79D2, and so I'm curious if they also produced a double knockout, but in this case, they're reporting something quite similar, that you can edit these biosynthetic genes and majorly reduce the cyanide content in cassava. So this was all well. We were excited to see that we could generate such a potent phenotype through genome editing, but there was a bit of concern when I joined the project about what the implications of the eradicating cyanide potential in cassava might be. And Juma et al. Also had this similar concern, wherein they report kind of more anecdotally that acyanogenic cassava might be more sensitive to herbivory. And they show that in their greenhouse, there are elevated levels of herbivory on their acyanogenic or their reduced cyanide plants. And so having this understanding, we sought to kind of look at the metadata that was available for cassava cyanide and see if we could ascribe any type of relationship between cassava cyanogenic glycosides and any type of yield. And so we were benefited from the beautiful data set that was made available to us from the cassava base and began to bin data according to cyanide level and yield. And what we found is that across any single way that we dissected the data, through geography or through time, we found that higher cyanide content was typically associated with greater yield. And in almost all the cases, we find that the low levels of cyanide or acyanogenic cyanide levels in cassava were typically associated with something lower yielding. And now, of course, it's hard to say that this higher cyanide content is causal in this case because we're looking across many environments and many cultivated varieties. But we did begin to believe that there was an association between cyanide in cassava and the capacity for that cassava to be higher yielding. And it was furthered by the evidence that increased herbivory was present in low cyanide backgrounds. And so collectively, we really began to understand that these CGs are playing an important physiological role in cassava plants, and that in certain contexts, their complete eradication would not actually be beneficial for yield and might not actually be what the growers need. And so we began to explore alternative options for how we might engineer cassava CG accumulation with minimal impacts on the overall physiology of the plant. And we looked to the previous literature and we found some exciting transporter work that described the action of transporters in plant functions to be able to move and mediate the accumulation of metabolites. So in this case, I was inspired by some early work that had occurred studying glucosinolate movement in Arabidopsis. And what these researchers had identified was that this GTR1 and its copy GTR2 were responsible for the accumulation and movement of glucosinolates within the Arabidopsis plants. So in this case, glucosinolates were being synthesized in leaf tissues and then being transported into seed tissues. And what the researchers in this study found is that through mutating GTR1 and GTR2, they could actually influence the accumulation of the glucosinolates in specific tissue types. So in this case, they were able to increase the accumulation of glucosinolates in the source leaf tissue through the knockout of these transporters and reduce accumulation in seeds. And I was inspired by this type of approach in the cyanogenic glycoside space. I thought that if we could identify and edit a transporter, we might be able to impact the accumulation of cyanogenic glycosides in root tissues, which are the primary food source that cassava provides. Of course, many do eat the leaves, as we know, but we thought that if we could increase the accumulation of CGs in the leaves, we might be able to actually maintain this herbivory or otherwise physiological role that the CGs are playing and reduce potentially the harm accumulated of consuming cyanide in cassava flour. And so we were benefited from previous literature that indicated that there was a shoot apex to root movement of cyanogens. And so we were excited to see that this data indicated that leaves were providing the source of cyanogens that were getting shuttled into the root tissues. And this indicated to us that if we could indeed identify a transporter, we would be able to potentially increase or decrease rather the root accumulation of CGs. And we were also really fortunate to have research that had putatively identified a cassava cyanogen glycoside transporter. In this case, they validated the function of this transporter in a heterologous system in a frog oocyte. So no one had actually tested this in cassava yet, and we thought, what a beautiful system and an exciting opportunity to use genome editing to edit the transporter that had been previously identified in the hopes of being able to introduce tissue-specific accumulation of cyanogenic glycosides. And so indeed, this is exactly what we did. You can see here on the left the edits that we introduced into the cassava plants. There were 2 unique genotypes, and each of those genotypes produced a knockout of the transporter, the MECGTR1 transporter. And our hypothesis in editing this transporter is that if the source tissues of the cassava CGs were indeed the leaves, we would overaccumulate CGs or cyanide in the leaves and reduce levels in the roots. And what we found actually really surprised us. We found actually that in our edited transporter lines, there were lower levels of CGs in the leaves, not higher levels, and that roots were basically more or less remained the same. And we also found that there were lower levels in the stems. So this was quite surprising to us because our expectation was that if the leaves were serving as the source tissue for the CGs, then we would expect that there would be greater volume of CGs in the leaves after we edit the transporter. But we found that the opposite was really true in our hands. And so we began to explore this further and did a similar girdling experiment inspired by the previous data that I shared. And what we find is that if we make an incision close to the base of the stem, where at the soil line, we can see that at that incision point, there's an accumulation of CGs below that incision point rather than above, which again begins to suggest that there's a root to shoot movement as well in these cassava plants, that perhaps it was the roots that were synthesizing the CGs in part and shuttling them towards the shoots. And so we took on our own girdling experiments, benefited by the edited events that we had of these transporters, to find that perhaps there is also this root to shoot movement in addition to the shoot to root movement that was suggested in previous data. And there was one further line of evidence that began to also further this this hypothesis, which was the expression data made available by this amazing cassava expression atlas that had been previously published. So we looked at the biosynthetic genes in these cassava expression atlases to determine where the biosynthesis genes were actually being expressed. And what we find is that the primary expression that is detected in these tissue types is actually in the fibrous root, which again supports this hypothesis that we were generating, that actually a primary mechanism of cyanogen accumulation and synthesis is in the roots, and perhaps it's getting shuttled upwards towards the leaves. And so this was true for CYP79D2, which is presented here, as well as CYP79D1. Both of these biosynthesis genes were found to be almost exclusively expressed at this developmental time stage in the fibrous root. And so with that, I want to just provide a summary of kind of all the data that we've accumulated in our cassava genome editing program. What we've learned to date is that cassava cyanogenic glucosides can be eliminated by CRISPR-Cas9 mediated editing of the biosynthetic pathway, and that the elimination of the cyanide potential could potentially have fitness impacts, partly mediated by herbivory response or some other physiological roles that CGs play in the function of cassava. And the folks on this Zoom call know better than me what some of those roles are putatively responsible for. We do have some room to play with the levels of CGs through mediated through genome editing. And so you can edit single copies of each of the biosynthetic genes to be able to impart a kind of intermediate phenotype. And through the work that we've done in transporter editing, we found that the transporter edited events had lower top leaf levels of cyanide, which provides evidence that there might be de novo biosynthesis of CGs in root tissues as the primary kind of source for cyanide within these cassava plants. And with that, I would like to conclude the research piece of the talk and just tell you a bit about a program that I've been involved with for the past 3 years and I'm an instructor for, which is our African Plant Breeding Academy CRISPR edition. This is a course that's based in Nairobi, but is open to all African scientists who currently reside in a country wherein there's a biosafety law available for the regulation of CRISPR-edited plants. And so every year we convene 3 sessions, 3 two-week sessions to train about 10 to 11 scientists from across the continent in CRISPR biology. everything from theory of how the molecules work down to designing a project that these students will bring back to their home institutions and actually get the opportunity to implement. And so you see here on the left, that's us in the banana greenhouse from our most recent session. The students are working on a genome edited banana that's got a disease resistance phenotype. And we're having a lot of fun with it. And the folks on the right is our second cohort, and they're off to doing amazing things. So I've provided a QR code there to our course website. We will be hosting a course this year. Registration will open in a couple months, but I want you all to keep it on your radar in case you have colleagues or somebody who you might be aware could benefit from this type of training. The application is limited to folks who have a PhD in plant molecular biology. This is not meant to be a plant breeding course. It's meant to be a CRISPR course. There's actually a separate academy for African plant breeders that's running as well. So I'd really encourage you all to explore the opportunities to share this CRISPR course with your colleagues. It's been an incredibly exciting time to see what CRISPR biology can do and offer in regards to agriculture. And with that, I'd just like to acknowledge the team that makes this work possible. The biosynthesis editing story was led by Jess Lyons and Michael Gomez, and the transporter editing side was led by Samantha Lieberman and myself, in addition to an excellent team of researchers here at the Innovative Genomics Institute and UC Berkeley. And I also want to acknowledge Cassava Base of NextGen Cassava for providing really excellent data for our meta-analysis, and the Konzo Prevention Unit for making the cyanide detection kits available. And with that, I'll thank you. And I think we'll take questions at the end.
Thank you, Nicolas. Unfortunately, I don't see the sign for clapping hands, but I'm going to clap here. Thank you so much for such a comprehensive report. Yes, questions will be at the end. We move now to Dr. Alex Ogbonna. He's a research scientist at a biotechnology company at the moment, and he focuses on seed design and more sustainable global food systems. His research focus on plant genomics, cassava breeding, and crop database development and deployment. He has led studies on the genetic control of cyanogenic glucosides in cassava to reduce dietary cyanide exposure. Alex, the floor is yours. You have 15 minutes or less to deliver your talk. Thank you so much. You can start sharing.
Thank you. Let me quickly share. Can you see my slide?
Yes, we can see. Perfect.
Okay, awesome. Thank you for the introduction, and I'm really honored to have the opportunity to share some of the work we did a few years ago. Hello, everyone, and thank you for joining us today. My research interest has been to leverage genomics for the purpose of expanding insights on domestication and diversification with the objective of improving crop adaptation generally. I think today I would be talking about the insight from a large scale historical GWAS we performed on cyanogenic glucoside content in cassava a few years ago. So one of the quotes that have inspired me on cassava was attributed to FAO, and it basically described cassava as the food of the poor that has become a multi-purpose crop that responds to the priority of developing countries, to trend in the global economy, and to the challenge of climate change. The projection is that in the next decade or two, cassava would be adopted by mainstream industries for the purpose of starch production, given its drought-resilient nature. Essentially, the objective of this work was to understand the genetic architecture and perform gene mapping of cyanide from breeding perspective. So I think previous study highlighted this inconsistency in the characterization of cassava varieties for cyanide content in Africa. And this has been attributed to the inconsistent transfer of ethnobotanical knowledge surrounding the use category management from South America to Africa. This resulted in post-introduction hybridization between groups, generally beta and sweet, not usually crossed together. And you can see this is shown in the bimodal and normal distribution for Brazilian germplasm, which we worked with, and African cassava, respectively. The Brazilian germplasm distribution is on my left, and then the African germplasm is on the right. So the inconsistent transfer of this ethnobotanical knowledge surrounding use category management from South America to Africa when cassava was introduced resulted in, change in the landscape of ISCN as a result of post-introduction hybridization between beta and sweet cassava land races in Africa. And so the lack of this differentiation between the two groups is highlighted both in the genetic variance and the trait heritability observed in the African germplasm. So we were fortunate to have access to essentially a good germplasm resource from Brazil that covers all the landscape of Brazil, but also we have access to 26 field experiment coming from 26 African countries, courtesy of cassava base. So what we try to do in the first panel here, we try to understand the germplasm that we're working with, did some characterization, and all of this are published. And then we looked at the trait architecture and also worked on developing some transcriptomic resources that can be used generally within the community to study gene expression of genes of interest. So what we did was to identify genetic correlation between cyanide content and genotypic variation. We perform mixed model genome-wide association mapping with Bonferroni correction as a test of significant SNP. 2 significant peaks were identified on chromosome 14 and chromosome 16 for Latin American germplasm. The idea is to understand what is happening in African germplasm, but from the perspective of Latin American germplasm because of the initial characterization that we've seen between Africa and Latin American germplasm. So based on optimal p-value, the candidate SNP we located within a mate vacuolar transport gene annotated as multi-drug and toxic compound extrusion protein. We also did a spatial genetic analysis, which shows that the allele driving high cyanide in this population coming from Brazil respond in a dominant manner and mostly concentrated in the Amazonian region. We also did functional annotation, which reveals a candidate SNP to be predicted to induce a missense variant. This mutation causes an amino acid change from threonine to alanine. It is predicted to be deleterious and reduce root cyanide by 30%. Now, after this result, we kind of went back to literature to check if the result we got aligned with the already described biosynthesis pathway. And what I showed is a schematic representation of the clustering of the cyanogenic glucoside biosynthetic gene in the genome of cassava. So the functional genes are represented by arrows indicating their orientation. So this study was originally done using cassava draft genome, which was version 4. So I looked up those genes in the genome version 6 and found those genes in chromosome 12. This was, as at the time this study was done, we now have more recent version of cassava genome. So to probe further to see if there is some sort of interaction between this region and this region we found in this study and the biosynthesis gene cluster. I performed genome-wide epistasis interaction analysis, and I found no interchromosomal epistasis interaction. Now, this leads us to suggest that, a distinct evolutionary path for HCN regulation in cassava compared to what was reported in sorghum. So I went ahead to look at kind of estimate a proportion of genetic variants explained across the genome and subgenome of interest. So chromosome 16, where we found our candidate site, and its candidate site explain 50% and 30% variance for HCN. That's the hydrogen cyanide as measured in cassava as a trait. So while chromosome 12 basically explain just 5% of genetic variants. In addition, I went ahead to, since there was no GWAS peak in chromosome 12, where the biosynthesis clusters were, so the question now arose, has cassava domestication, targeted upstream or downstream genetic regulation steps of HCN biosynthesis. And what I did to answer this question, I compared cultivated cassava to its progenitors using HapMap2 whole genome sequencing dataset. This was the data set available that captured genetic diversity, global genetic diversity for cassava. So the plot here shows the distribution of the SNPs markers that represent fixed or nearly fixed differences between Esculente and Flabilifolia in the HapMap2 whole genome sequencing dataset. The legend scale indicate the number of SNP within 1 MB window size. So the x-axis, the y-axis are the chromosome, and the x-axis are basically each SNP within each of those chromosomes. So the purple legend represents the identified region. The purple star, represents the identified region for cyanide regulation in this work, suggesting that domestication can impact metabolic content targeting transport regulation. And this has been reported earlier in maize and in rice. So in view of this finding, we speculated at that time that cassava domestication may have actually specifically targeted downstream genetic regulation step of HCN biosynthesis. This is also supported by the fact that root size and HCN content are the major traits of cassava domestication, as highlighted by Ramu in his previous research. Now, so to characterize this candidate protein, I carried out a protein sequence alignment of mate genes in cassava, in sorghum, and in Arabidopsis. Now, this protein alignment and comparative phylogeny show close sequence homology between the GWAS candidate gene and SB-MATE2, a characterized vacuolar membrane mate transporter in sorghum. It functions and is highlighted in this particular region that is in red rectangle. So it functions in the accumulation of plant specialized metabolite such as flavonoid and alkaloid. So this SB-MATE2 in sorghum has been reported to export DRIM, another hydroxynitride glucoside. So we started functionally characterizing this mate gene with Miguel's lab, a USDA lab at the Cornell campus. I think we got slowed down a bit, but hopefully we would complete that work. So like I mentioned, we try to understand the germplasm coming from Africa, but from Latin American perspective, given that, there have been some hybridization that has in terms of generally modern breeding has, post-introduction hybridization that did not separate between sweet and bitter cassava as is obtainable in Brazil. So we kind of did GWAS within African germplasm, and the result was similar to what was observed in Brazil, and that's the lower Manhattan plot. So we also looked at, joint GWAS between Africa and Latin America, which further confirmed our finding. And one takeaway is that in both data sets, ISCN showed this conserved genetic architecture. One additional thing we did to ensure that the current study captures the diversity of HapMap2 panel, which was the data set that represented cassava diversity at global level, we generated this mate candidate protein sequence from the cassava HapMap2, 241 assertion. We did alignment and a maximum likelihood phylogenetic tree, which is shown here. And we using those sequence for the candidate protein for all of those individual 241. What we found is Manihot fablifolia individuals are preferentially homozygous, which are the red, the samples that are colored red, and they have high cyanide for that candidate candidate protein, which is in line with its phenotypic characterization for HDN content. Now, interestingly, we found Manihot glazovia and Manihot prunosa individuals gene sequence are all, homozygote AA, which is low cyanide for the, and they also clustered separately from the flablifolia, which are the green individuals, colored individual. The black are heterozygote site for the candidate SNP. Just to summarize and what I've discussed here, or what we found in this study, is that HCN showed conserved genetic architecture. Both data sets shows that HCN variation is regulated in an oligogenic manner, looking at data set coming from Africa and that of Latin America. We also looked at data coming from SIA that we had in cassava base. I think the result was consistent. So evidence in our current data set shows no identified epistasis between this chromosome 12 that has the biosynthetic gene and the regions that we found in chromosome 16 especially. And we also saw that cassava domestication targeted downstream genetic regulation steps of HCN biosynthesis. We saw that the major loci allele for high cyanide in Manihot esculente seems to originate from its ancestor, Manihot flablifolia. And lastly, we saw that the co-domestication of sweet and bitter cassava major alleles are dependent upon geographical zone. In terms of deliverable, we were able to develop and validate CASPR marker that is available for the community, but also this marker set, serve as fast and cost-effective molecular diagnostic toolkit for breeding purpose to increase selection efficiency. I believe they're used in breeding programs in Africa and in Brazil. A lot of people contribute. A lot of people contributed to this work too numerous to mention. And this was a picture from August 2021 of Mwile Lab, where I was working at that time, was doing my PhD. And thank you for listening. Hopefully I would have time to take some questions.
Thank you, Alex. Good presentation. Thank you. Next speaker will be Dr. Kirsten Jorgensen. She's associate professor in the Department of Plant and Environment Sciences at the University of Copenhagen. Her research focuses on understanding plant physiology and the role of bioactive compounds in plant responses to environmental challenges. Her main research aims are to uncover how plants adapt and defend themselves, and to translate this insight into practical tools for sustainable agriculture and resilient crop development. Kirsten, the floor is yours. You are muted. Sorry, Kirsten.
Sorry, yes. I hope you can hear me now.
Yeah. We can see your slide very well.
Well, that sounds good. But thank you for inviting me, and thank you, Carlos, for telling me that there's nothing like old science, but there's only science. I started working on cassava actually 25 years ago. So some of this is the journey from cassava, from that time until now, and the observations. And it's good to listen to Nicholas and at least come nearer to some of these things which we observed through these 25 years. If you have been asking me, and now I can't, sorry. If you've been asking me 25 years ago, we have somehow told you that actually cassava and synthetic glucosides, that's a defense compound, and that's what it is and nothing else. But through all the observation we've had, we've found out that it's much more than just defense compound, but is also involved in the development of plants and also reaction of the plants as such. So it's much more than just defense. So I think we said before that synthetic glucosides are actually quite ancient biomolecules. It found a lot of plants by now. Sorghum has been mentioned, cassava, lotus, almonds, clover, apples, barley. And in cassava, we will find mostly linamarin, but also lutostolin for these ones. So actually, we started 25 years ago. We wanted to, as many others, and Nicholas also wanted to obtain an acidogenic cassava, a cassava with less or without any synthetic glucosides. So that's why we set out to develop a genotype independent procedure, because we wanted to have transformation procedure which we could use any elite type as such. So we got a lot of African elite lines from ITA, and here's just some of them, and we just state here the transformation frequency. But compared to Nicholas, we actually chose the other method, which is for cassava. We chose the one which is based on Lee et al. From 1995, which is using actually making cotyledons and not having a color step. We find this much more robust because we are not having a liquid media, we have it on solid media all the way through, and we induce the small cotyledons on these ones, and then we can keep it actually in culture making cotyledons, and it's the cotyledons we cut and actually use to transform with agrobacterium. And here just to show that that's how the cotyledons look in different cultivars. And actually, maybe you can recognize that this 1371 is one with the cannabinoids in the ones. It's much more yellow than the other ones. And we are using one of the South American lines, MCOL 22, which our first generation is based on. So looking at the synthesis of synthetic glucosides, we through these years actually also investigated and found the different steps in the synthesis. So actually there's two alleles and different ones, the D1 and D2. And the 71, we have E7 and E11. And the UGT, we also have two of these ones. And what we saw actually when we investigated the synthetic glucosides, that's a very high variation of content through the different clones actually. So just here showing in the different colors, there's different clones of the same cultivar, and actually sometimes it differs 50 to 75% of the content. Of course, we need to bear in mind we are in Denmark. Denmark does not have a tropical environment, not even in our greenhouse, but still it's under consistent circumstances. So we just were curious to know how is it that the variation is so high. So the next step was actually to look into making this acidogenic cassava, and we went for a knockdown and knockout. At this time point, the CRISPR-Cas was not kind of invented yet, so we're using RNA interference instead to look at these ones. The first generation was with the 35S promoter, and you might can see here from Chang et al. That 35S promoter might not be that good in tuber as compared with the native promoter of CYP71. So the next generation, we're using native promoter instead in front of our RNA construct. And we had some observation of the transformants from these ones. Looking at the first generation, actually we could see in the leaves and the tubers, we didn't have the same downregulation. So actually some of the same, Nicholas also stated that there might be a transport from the tubers or tubers actually making synthetic glucosides as well on top of what's made on the leaves. So there might be something in the transport, because you see here, we have the lesser amount here in the tubers, but you cannot really see the same pattern in the corresponding plant's leaves. Then there might be some regulations different in the tubers and leaves. And we saw in the stem, the transcripts are found in the paramagnetic cells around the xylem, the phloem, and actually also the cortex. So these could be regulated differently, and that could also tell something about the transport up and down. And then for the first generation, we knew that the 35S might not be as good in the tubers, so the downregulation in tubers might be different due to this one. So looking at downregulation, this is still the first generation. We actually saw phenotypes in vitro. You see that A and C is 2 transformants and the wild type in the middle. So they got long and slender, have long or short, thick roots for these ones. But as soon as we went into the greenhouse, either pure sand, soil, they look just as a wild type. And looking at the tubers, they look the same. And again, we're in Denmark, the tuber size is not the same as the rest of you have shown, but at least we got tubers we could investigate. So the second generation where we have the native promoter, and that's why I think Nicolas' experiments are really, really nice, but using a knockdown, we saw that all our transformers went to more or less this stage with the 2 small leaflets, and then they stopped growing, they turned white, and actually they died. So that set out the thought that these compounds also might need to be used for development, especially on this stage, because another one saw that the production of cyanidin glucoside started when this plant was this size. So that also made us to investigate what about the free amino acid in the tuber tissue when we have the transformant. Look at the first generation, there was no difference. But looking at the second generation where we have the CYP71E7 promoter in front of the RNA interference for the CYP17 9 D1 and D2, we could see a very strong difference between actually the control 1277 and then the one of our transformance plants, which are the yellow bars of these ones. So actually it affects the amino acid, which we actually would also have predicted. So the next we looked for that, because we actually saw all these variation in our data, and also we found out actually that there is a diurnal variation in linamulin through the day. And especially if there's a high light intensity, we saw there was a drop in actually content. And we measured it several times through the whole 24 hours. And one thing is looking at linamulin content, because the turnover might not be that fast, but looking at the transcript, there is very pronounced, that you could see just before the night, there's a drop in the synthesis gene of the 79s and 71. The UT did look a bit differently, and especially they had 2 different patterns. The 85K5 also have a drop just before the night, where the 85K4 actually looked a bit different. It was more uniform compared to the other one. The same pattern was seen when we looked at the protein profiles. And here we looked at CYP71 and the CYP89 and the linamulase. We actually saw that there was a reduced in light, and there was another profile during the night. So somehow the content of cyanidin glucose are also affected during the day and also during high light intensity of these ones. And I kind of confirm Nicolas about the if they're more prone to being attacked. We had a mealybug attack in our greenhouse, and they loved our acinidinic cassava plants, and they were really much eaten by the mealybugs. So this next step we actually went for was actually look into how are we going to improve the nutritional value of cassava. And there we were looking actually to use potassium because it was well known from potato. So the thought was that does not need to have any toxicity proven for these ones because it's eaten all the way today. And we had one which was generally inserted, one which was accumulated with the starch granules, and one was associated with the starch as such. And looking at these ones, we actually saw that the first assumption we had when we started to make cyanogenic glucosides decrease would be that it might be affected the protein content because it would use less amino acids, and we also saw the pattern of the amino acids was actually affected. But it was not the case. We didn't see a rise in protein content, and we also saw that our plants show this severe phenotype in vitro. But however, when we inserted the gene encoding patatin, we saw that in the tubers, we actually also decreased the content of cyanogenic glucosides. And I'm sorry to my technicians, but I thought they had mixed all our samples around, but we repeated several times, and we actually saw that increasing patatin, increasing protein, actually decreases content of cyanogenic glucosides. So there might be another way of affecting the content of cyanogenic glucosides. So I would just want to say thank you to share some of our observations for the cyanogenic glucosides having more functions than just being a defense compound. And I also want to thank you, all the people which has been involved. There have been a long range in the group of Professor Bill Lindberg Müller during these 25 years. And we were supported by a long range of funding from Willem Foundation to Danida, and also thank you for IJ actually funded us for 3 years as well, and our Danish National Research Foundation as well. So thank you for listening.
Thank you, Kirsten, for your presentation. Very nice. The last speaker is Dr. Hernan Ceballos, who earned his PhD in plant breeding from Cornell University. And he worked as a maize breeder at CIMMYT for 7 years. Hernan also worked at the Universidad Nacional as a professor in plant breeding and quantitative genetics. And he is very well recognized as a cassava breeder for more than 20 years at CIET. He has been distinguished with several awards, and he's a good friend of mine, that's for sure, Hernan. The floor is yours, Hernan, to start sharing. Hernan, do you hear me?
Perhaps he's on mute. Hernan, are you on mute?
I think he's having trouble because I have asked him to unmute and he has disconnected automatically.
Maybe he will connect very soon.
Yes.
There's a comment in the chat from Lynette Nasiroli asking about information from the African Plant Breeding Academy CRISPR course. It was provided up there by Isabel, but we can copy again that. There it is. The information that you are asking for is there in that link. Okay, please open it up. Okay.
Yeah, and if I can jump in on this as well,
Sure.
just to comment, is that this is why we established this network and this working group, is actually to make the knowledge available. The reason we're having this symposium is so that these amazing researchers, whether they're working on Konzo or they're working on cyanogens in plants, that we can have these put together, brought together, and recorded so that we can spread that knowledge. And then the session tomorrow is going to be about food systems and public health. So I just think it's, this, Lynette, this question of yours just shows exactly why this network is needed. You can join the network, it doesn't cost anything. And so we've got a smaller group, which we're just calling the working group, which we can, where we are holding, we're organising things like this, but it's really all about the network and putting people together from different disciplines. So I hope that is helpful to people to know that that's why we're having it, and that there is a great need to understand more about cassava, to understand more about the relationship with Konzo, and so that's why we have it, and to also continue the work that was started by, you know, Hans Rosling, Julie Cliff. Julie was on the previous session. I don't think she's with us now. Bania, you know, there's so many important people who've done work that we're in danger of losing because some of those people who've done the pioneering work. And I think this has been a fantastic session to see the journey from Kirsten's pioneering work of working out the pathway through to how it's been knocked out now. So how are we going with it? Anand, have we got him online yet?
No, not yet.
I think while we're waiting and Carlos is engaged with that, we can follow up. Kira has now put in the chat that you can email her, or you can also use the, there's a generic info at International Safe Cassava Network email. Kira, maybe put that one up too. Yeah, people can do that. Now, while we're waiting for Anand, why don't we have a few questions? Does anyone? I've got lots of questions, of course.
I don't know if it's me, but I don't see any questions in the Q&A. Is it because I
cannot? No. I think we can just ask people to see. We could ask people to unmute if they haven't done that.
Yeah.
But I have it just while, you know, do you think Hanan is going to get on?
Hold on a second. He's calling me. Hold on. Go ahead.
Okay. So I'll ask a question of Nicholas while we're waiting. Of course, I'm very interested in this thing. It was actually first after talking to Kirsten years ago when you were talking about your acyanogenic transformates. You know, some of them didn't survive until you put them in soil. And then talking with colleagues both in Mozambique and in Fiji that the more bitter cassavas, the high cyanide ones seem to yield better. Nicholas, you talked about that as well. Why do you think they are yielding better? So do you think it's just the herbivory, or do you think it's maybe like what Kirsten suggests and there could be some physiological benefits?
Yeah, I definitely agree wholeheartedly with Kirsten about the overall physiological role of CGs in cassava physiology. I think it's easy to point to insects or herbivory as the primary kind of role that CGs are mediating in cassava physiology, but I think that they're playing a much more interesting and overall very ingrained physiological role in cassava. I think they can be serving as sources of nitrogen. They can be remobilized in specific environmental stresses. We know that they are involved in many processes besides herbivory. The extent to which they're influencing all these diverse roles is harder to assay. But now that we've got this type of edited germplasm, I think there's a really nice open opportunity to begin to ask and answer some of those questions. And so I'd be excited to see the community continue to explore the kind of biofunctional role of CGs.
So I got news from Anand. His computer decided to reset after he unsuccessfully tried to unmute. So he said give him a few minutes to get back online, okay?
Okay, so if we can...
yeah, I do have the presentation too, Isabel. I can share it, but he's not able to join yet. So
yeah. So Kirsten, that's great. So we can just continue with this part of the discussion now instead. Yes. Kirsten, I wanted to ask what your comments were on this.
Yeah, I think actually for the function of the sunlight in cassava, I also think about the work we've been doing at the sorghum and with the seed. When you actually lower the content of glucosides, that the seed might germinate worse than the other one, and I could compare it with some of the results we see. And then I'm just inspired by Nicholas' talk as well of when we look at where the transcript for the census genes are found in the stem, it actually could explain some of the things with the transport up and down, because it's both around the phloem cells, the parenchymatic cells and phloem, the xylem cells and the cortex. And we always wondered why we actually see it in kind of free bands. But they could actually explain some of the different functions of the Sandiango glucosides, and also explain why when we girdle, sometimes when we're on top of the plant, we see something, and the bottom we'll see something else because the trends would be different in these two places. So I think there's a lot of things we can start to connect with all these new results coming up. Instead of in old days, it was just a big question. And that's still something I wondered when we made it, that when we make the test for Sandiango glucosides in plants, it differs if we take 10 milligrams of tissue in one tube and 3 tubes of have 30 milligrams, you get different results, even though it's the same leaf. So that is a regulation in the cells. And if you boil it, you get the same results in all of them. So it's not, we've not cracked the regulation of Sandiango glucosides in the degradation in cassava yet. And I think still some of these really open questions. I really would love to, if anyone could answer these ones. How is it regulated, act on cell basis, even when you take leaves out?
Yeah.
I would love to just add one more thing to follow up. One of our data points that really surprised us was in the transporter knockout. We see this difference in leaf accumulation, so lower level and top leaves. But then the roots, there is no difference, not a significant one. So it suggests to us that there is some type of sensing that the roots are taking on at the extent of the CGs that are present, and some auto-regulation to the max extent. So this isn't just about biosynthesis, this is about CG sensing, and the certain varieties have the capacity to be able to accumulate more CGs. Their upper threshold for detection is different. And so we were, and I just want to add one final thing is that we've produced the knockouts in sorghum as well for the Durham biosynthesis pathway. So we'll be excited to chat more about the potential of these types of lines.
Yeah, the same regulation we see in leaves if you make these ones. And also when we look at our Sandiango cassava, the downregulation in leaves is not the same as roots.
We have a question. Sorry, we have a question from Desiré, who's has the hand up. Please go ahead.
So I have a question to the panelists. Now, have you thought about, so what would be the implication of identification for everyone on the panel? What is the implication of your work in terms of applicability when it comes to grow cassava on the field? So when I look at soil conditions in the southern part of Congo, for instance, I'm wondering if it would still be possible to even get some of the levers that we are talking about to be grown on the field. So what would be the applicability of the findings that you have when you take into consideration the soil condition and water issues, and water stress?
Someone want to answer that? So what are the implications of this research, for example, for African smallholder farmers and also the industrial level in which is happening in some countries? I think there's two different questions there.
Yes.
I can take a first pass at the question. So I ended my talk on the African Plant Breeding Academy because it's one thing to do this fundamental research that empowers understandings of cassava. It's another to actually translate that research into applications. And I was so inspired by session one to see, the boots on the ground work in developing interventions, really effective interventions. Our vision for the African Plant Breeding Academy, of which many of our graduates are actually going on to do genome editing in cassava, is to empower scientists at public centers. So they're at NARS or at national universities doing their work. And so we're hoping to bridge kind of these fundamental understandings of cassava CG physiology with real impact in the field and translational work. The reason that I really emphasize that one single gene knockout intermediate phenotype as an exciting phenotype is that we're learning a lot about how acyanogenic varieties might be, very susceptible to herbivory or not the most suited for the field. But even through these kind of early studies, we've learned a lot about how a single genome edit could translate to something that has some agronomic utility. And so the hope is that we can collaborate across the continent in hopes of actually seeing some of these fundamental insights get translated to growers. And as part of the African Plant Breeding Academy, every single one of our students is linked to a team of breeders. They're required actually as a graduation requirement to work directly with breeders to bring a value-added trait into a product and actually deliver. And so we're tapping the infrastructure that already exists in plant breeding, in cassava plant breeding specifically, to make good on some of the translational aspects of this research.
So if I may add to that, hello, can you guys hear me?
Yes, that's
Okay,
good. Go
cool.
ahead, Alex.
Yeah, so if I may add to that, to what Nick has mentioned, I think generally from breeding perspective, from our side, what we try to do is to take the forward genetics approach and to develop some tools or marker set that breeders can use to select, cassava varieties for different use cases, whether intermediate or low or high cyanide, at least to improve breeding efficiency in that sense. And currently, this markers are used in ITA, at least I know in Bergen, and also they've done some work to validate it independently, but also it's also used in Brazil. So I think this is a low-cost breeding tool that can be used to improve the selection efficiency for this trait within a breeding program.
So, Ross?
Yes?
We're going to try the following. I'm going to share Hernan's presentation. I have Hernan here on WhatsApp. Hernan, can you say something and see if we can hear you well?
I am for this.
No, you cannot hear?
Try again.
We hear sounds. I mean, we hear his voice, but we don't understand.
Oh, really?
Yes.
Yes, we know
Yeah,
it's him, but we don't understand the words.
yeah, probably it's a lot of interference. That's what usually happens. So he's having still problems getting his computer up. Okay. So one option could be that we have him record the presentation and then we post it as part of the symposium, and then the questions will be emailed to him. Okay. Would that be okay?
Yes.
Yes.
Yeah, I think we could do that. I think the other thing we should consider, Carlos, is that we could invite him to give one of our webinars next year as well. So he could give a longer half-hour presentation at one of those. So that would be a backup option. And in addition, not instead.
I don't know if you hear that, Hernan.
And what I can do is connect tomorrow. I'll be able to join again and talk to Isabel.
Okay.
See if I can record.
Or the other option that Ross mentioned is that you present a webinar because we meet every couple of months. Okay, that will be another option. Okay, thank you so much. Don't worry. Okay, don't worry. You're part of the network. Okay, you are hooked into it. Bye.
An important part of the network.
Yes. All right.
He's the guardian of all those different cultivars.
So we have about 10 more minutes for questions. Somebody had a hand up. It was Alex or who was it? No?
I wanted to essentially contribute to the question asked by Desiré, which I did. Yeah.
Okay. So I do have a question for Nicholas. In that Plant Breeding Academy CRISPR course, has there been any interest or projects for editing to achieve a low cyanide cassava in Africa?
There hasn't been a low cyanide project yet for one of our graduates, but we have trained students who are working on cassava trait engineering using genome editing, but not a low cyanide variety. I think some of our preliminary data has begun to suggest that despite being able to produce this trait it might have some detriments in terms of agronomy. And so because our students are required to work with plant breeders, the plant breeders actually recommended against the use of this trait. And so it hasn't been adopted from any of our students, although they're working on very exciting cassava traits. And so I think from the fundamental side, we need a little bit more understanding of how best to select targets such that we might generate cassava varieties that perhaps have an intermediate phenotype or are less susceptible to some of the negative pleiotropic impacts of removing CGs.
Sounds good. Any other questions from
I have another question, but I
Ros?
can't dominate. Please, anybody in the participants, I think if you can't work out how to use the Q&A tool, just put it in the chat or just put up your hand and indicate.
But go ahead, Ros.
So I had a question for Alex, and I was interested, is this correct? The overall, you have this broad spread of plants with cyanogen levels, both in the Latin American varieties and in African varieties, but you have more of the bitter ones in the African data set. Is that correct? And then, no.
Go
maybe
ahead.
I missed that.
Sorry,
No, so
ahead.
I was just wondering if there had been selection inadvertent by farmers, like selecting for high cyanogen levels in Africa because it may have some benefits in yield or tolerance to stress or something like that. But I may have misinterpreted your data, so please.
Yeah, thank you, Rose. It's actually a great question. I think I didn't look at, kind of essentially compare if we had more high cyanide variety in Africa compared to Latin America. What we did look at is if you look at different regions in Africa, we had data coming from West Africa, we had data coming from East Africa, Southern Africa, and Central part of Africa, essentially covering sub-Saharan Africa. What we saw is that in Southern Africa, we had higher materials that had higher cyanide content compared to West Africa and East Africa. But East Africa is also slightly higher compared to West Africa as well. So if you look at it from the perspective of different regions within Africa, you have Southern Africa and East Africa to have higher cyanide content compared to West Africa. Yeah, I think between Latin America and Africa, we're just looking at if modern breeding has impacted the trait architecture of the cyanide content in Africa, because we have this post-introduction hybridization that was generally done in Africa, where they don't have this use knowledge, information regarding the sweet and the beta. So generally what they did was to cross beta and sweet cassava together. But in Latin America, especially in Brazil, they kept this breeding program separate. They mostly, make crosses within sweet cassava and within beta cassava separately. And you can see that in the previous report for those different materials coming from those different regions.
Thank you.
Yeah, the other interesting fact somehow is that there has been integrations from the wild in Africa for virus resistance, and I wonder if some of that has influenced cyanide.
No, we didn't see that. Why we saw that that introgression, and I think we also reported some past introgression that we found in Latin America in one of our publications, but we didn't see a direct correlation between that introgression to African to high cyanide. Rather, what we saw is the role cassava domestication has played in kind of moving in terms of the study which we did to map the gene in terms of moving, having more downstream impacts on cyanide regulation, because we didn't find the regions and genes that we found were not, in any way interacting or associating with the biosynthesis gene cluster. But this is from breeding. This is more from the approach of forward breeding or forward genetics as compared to most of the work done by Nicholas that is based on more reverse genetics approach.
Okay. I just want to check with Desiré if there's a lingering hand there, but your hand is still up. Is it a new question?
I have a question.
Okay, go ahead.
Yeah, so it's a quite provocative idea, because we talked a lot about negative effect of cassava and Inari, but we also know that there may be some benefits of having cassava, not necessarily as a staple, but as a crop or within a diet that can bring to us some kind of benefit. There is this story, for instance, about impact of the cassava diet on cancer, for instance. And within our network, we have people looking at prevalence of cancer in cassava-reliant populations, because we know from experimental studies, either in pheochromocytoma, PC12 line, or glioma cell lines, you get linamarin getting into a cell using a glucose transporter, and once linamarin is into a cell, it's break down and produce cyanide, and cyanide kills cancer cells. So maybe the whole idea is not about having a cyanogenity varieties of cassava. Maybe the whole idea is about, because even when you look at things from the toxicity standpoint, everything is about dose and concentration. Even water, even Coca-Cola will kill someone if you get over threshold. So maybe the problem is not linamarin. The problem may be the whole issue of to which extent a single person and in which context can tolerate what comes. So I know it's a difficult question, but I just want to make sure that
maybe a good topic to have a webinar. I mean, it's a valid topic maybe to bring some people for discussion. Any reactions in the next couple of minutes that we have to close this session?
The only thing I would say that linamarin is actually, you can buy it as an anti compound in health shops.
Mm-hmm.
So that could actually support your idea.
Yeah, for sure. Okay, Ros, there's people there for whom it's almost midnight, and there's us that needs to have dinner. So any words of wisdom before we close this session and reflect for tomorrow?
No, there is actually a question in the chat.
Is it?
And I think Desiré has in part answered it. It's like, why are people still eating these cassava varieties while they're known to cause Konzo or higher levels?
Okay.
I mean, does anybody want to answer that? To me, it's like, it's the most amazing crop for how much you get per hectare on such terrible soil with no fertilizer. It's got to be a great crop.
I have an answer to that. That was my first question 30 years ago. I've asked people in a village when Tylleskar has introduced me to the word Konzo. I've asked this elementary school teacher this exact same question. This is what he told me. Because we do not have anything else to eat, we prefer to grow bitter cassava so that no one will steal it until we get to harvest and make cassava flour. And that was puzzling to me. And that was the moment when I learned to understand what Professor Hans Rosling used to say, that poverty was not lack of something, it was a lack of choice.
Yeah.
And in those areas, there is no choice. Poverty is an issue, and it's a disease. And they've learned from that. There's just no choice.
Yeah.
The soil is very poor, and there's nothing else to grow. And they prefer to rely on that. Mm-hmm. And I remember FAO in Congo, in Kahemba, for instance, close to Kahemba, there are different crops that are brought in by FAO, but people have used them just for a few months, and then they just decide to leave them, to drop them. They prefer to grow the bitter one so that no one will steal it.
Yeah.
Yeah, I think that answers Poas's question about, you know, can we just provide bitter cassava, a sweeter varieties, because there is some choice in planting bitter ones, and also, of course, there is some evidence of higher yield. I think we've suddenly got lots of questions, but the time has come to an end, so I think we're going to have to finish. I have questions for everybody here. If anybody wants to test these varieties, say the effect on drought and recovery, Kira has perfected the method, but we didn't get a very exciting result, so we're going to publish that as a method paper. So we can provide that method to you to actually test this in a very controlled manner. So just to finish, I just want to, of course, thank Carlos for the speakers in the session, Thorkild and the speakers in the Konzo session. The Konzo Prevention Fund was set up as a memory to Howard Bradbury at the Australian National University, and there's a very small amount of money coming from that to support the Safe Cassava Network and the Safe Cassava Working Group, which has arisen out of the now folded Cyanide in Cassava Diseases Network that Howard Bradbury set up. I think we have a lot to thank these other people for. The aim of the network, as you know, is to bring together people in plant science, agronomy, food science, biotechnology, people working on cyanogenic glucosides, Konzo, and food systems. And so tomorrow, the session that we have is going to be, the first one is going to be on public health. That will be at 8 o'clock in the European time, 8 p.m., 6 a.m. here in Australia, with speakers from Mozambique, and also talking about the World Health Organization recent review. And then the one after that is going to be about food systems, chaired by Louise Abayama. So with that, I would just like to thank everybody. You're welcome to make a donation if you want to, but as I said, you do not need to make a donation. Let's continue the conversation. Let's have more network meetings. Let's have some more webinars where we can really dig into some of these issues. I think it's really important. So thank you, everybody. And I'll hopefully see all of you and more tomorrow morning talking about the public health issues. Thank you.
Bye-bye.
Bye.